Okay, good afternoon, everybody. I hope you had an enjoyable lunch break. And we're continuing with the third session of this great symposium so far. I'm Martin Nalijn. I'm chairing this session, and I'm in the University of Groningen, the Netherlands. So for those people who are also in Europe, like me, good evening. And we'll continue this session with talks about COVID-19. And we'll start off with Kerstin Meyer, who's in the Sanger Institute, and who is very well established in single-cell sequencing, especially of the lung, and also very heavily involved in the Human Cell Atlas. And with that, I would like to give the floor to Kerstin.
Okay, Martin, thank you very much for that introduction, and also thank you to the organizers for giving me the opportunity to present my work here today. And I will be talking about the cellular landscape of the upper airways in children and adults with COVID-19. Now, you're probably all quite familiar with this a portal from the Johns Hopkins University that charts the number of COVID cases around the world. And we are sadly reaching now this milestone of almost being at 1 million global deaths. And I think the numbers speak for themselves, but really, it's really a call to all scientists around the world to do whatever they can to help in understanding COVID better and coming up with possible treatments to help with this disease. As I'm sure you're also all aware, COVID-19 is caused by the SARS coronavirus 2. And actually by now we do know quite a lot about this virus. It is covered by an S or spike protein on the surface of the virus. And that spike protein binds the viral receptor, the cellular receptor ACE2. Now, in order for the virus to be internalized, the S protein needs to be cleaved by a protease, and TMPRSS2 is one of those proteases. Once it's internalized into the cell, the viral genome is released, and you can then get viral replication. Now, with knowing that ACE2 is the viral receptor and that TMPRSS2 is a very critical protease, it occurred to us that the Human Cell Atlas was actually a great resource to better understand exactly which cell types can be infected by this virus. So we actually made use of existing data sets and asked which cell types express either ACE2 or TMPRSS2, or indeed both, which is the relevant combination. And what we found is that in the nasal cavity, it was ciliated and secretory cells, and actually in the lung and bronchi, in the bronchi, it was those same cells, but expressed at lower levels. And then in the alveolar and parenchyma, it was actually the AT2, the alveolar type 2 cells, that had relevant expression of those two genes. And all of this work was published by Signac et al. In Nature Medicine earlier this year. And the paper actually gives a much greater overview of other organs and the cells that can harbor that have this receptor expression. But today I just will focus on the upper airways. Another thing that is possible with the data from the Human Cell Atlas is, for example, to compare receptor expression and then ask whether that tells us anything about transmissibility. So what this slide shows here is a number of different viruses here, SARS-CoV and SARS-CoV-2, and they, of course, bind ACE2, and then here is the expression that I've just shown in the different slide. And we can now compare that to a number of other viruses, like another coronavirus here, influenza and MERS, which bind ANPEP, STGAL1 and 4, and DPP4 respectively, and compare expression. And actually what we found is that those viruses that had a very high R or transmission value also showed very high expression in the upper airways. Whereas actually MERS, which has relatively low transmissibility, you really see that expression here is much lower in the upper airways and higher in the parenchyma. Now, I'm sure this is not the only story that governs transmissibility, but it's very interesting how we can harness data from the Cell Atlas to start to get an idea about the biology of the virus. We also looked at genes that are co-expressed with ACE2 and found that, so looking at the different cell types of the lung and then asking for co-expression, we noticed that there was a gene signature that was very strongly enriched in innate immune genes that are shown in red here. And other people have taken this work further and actually shown that ACE2 is a gamma is an interferon inducible gene, but again, it already tells us something about the biology. But what I'd really like to do today is, so all of that was published work, of course, but I'd now like to come on to the work that I've been doing with studying COVID-19 in children. Now, the good news is that in children, COVID is much less frequent, and actually only 1 to 2% of the global cases are reported in children, and those children that do get the disease have much less severity. And deaths are actually very rare, as shown in this diagram here. So that leads to the questions, why is COVID in children less frequent, and why is the disease usually less severe? And in order to start to address those questions, we wanted to profile the cellular landscape of the airways in healthy children and COVID-19 patients. And this work is actually a very close collaboration with Marko Nikolic, who is based at UCL in London, and our group at the Sanger Institute. And here is the patient cohort that we've assembled. Of course, we set out to initially collect about the same number of COVID positive and negative cases, but by the time all our labs were ready to go, the pandemic was actually beginning to come to an end, at least the first wave. But we then continued to collect COVID negative children, and now have a cohort of 38 children where we have a large number of nasal and blood samples, slightly fewer tracheals. And we also have 4 COVID positive children that actually cover different age groups quite well. And we then collected a number of adults as comparison, and also a number of convalescent adults who were COVID patients with severe disease 12 weeks after discharge from hospital. Now, for all of these, we collect the airway samples, which were brushes. They were freshly processed by cold digestion and then loaded onto a 5' 10x reaction platform. And we also sequenced TCR and BCR. At the same time, we also collected matched blood samples. These were processed somewhat differently. We used FICOL to identify PBMCs that were frozen, and then we could thaw them and multiplex them to run them on a 5' site-seq panel with about 192 antibodies. And again, TCR and BCR was able to be looked at. But for this study, the sequencing is still in progress, so I will only talk about the airway samples. And here's the data set. In total, we have about 150,000 cells, and in this relatively coarse clustering, we see 27 different cell types. And just a slight disclaimer here, this is a very fresh data set, so the analysis is still very much in progress, but I'll tell you what we know so far. So we see all the cell types that we expect. So down here, we have a number of different basal cell types, and then we have a number of different secretory cell types. And in blue here, we have the ciliated cells, and that's the epithelial cell population. We then also see immune cells. There's a B cell cluster up here, a T cell cluster down here, and this is a macrophage myeloid type cluster. But one thing that really struck me when I looked at this data is we've looked at quite a few different lung data sets, and usually the secretory cells and the ciliated cells map very separately and are not connected. Whereas in this data set, we see this population here, and we refer to it as inflammatory epithelial cells for reasons that will come obvious in a moment. And interestingly, this data set, when we ask whether these cells come from COVID negative or positive patients, the vast majority are from positive patients. So it really seems to be associated with disease. But we didn't just want to do this by eyeballing. Natsuhiko Kumasaka in the lab carried out a statistically rigorous analysis of the cell type proportions, taking into account all the different clinical metadata sections that we had, and asked what is different in current COVID patients versus non-COVID patients or past COVID patients. And first and foremost, we see that this inflammatory epithelial cells is upregulated. So we were very excited to see that this novel cell state does indeed appear to be associated with disease. We also see monocytes and neutrophils upregulated, and that's very much consistent with other people have reported, for example, in bronchoalveolar lavage fluids. We also see that B cells and T cells are downregulated, and even though this is in tissue and not in blood, I think this mimics the lymphopenia that again has been reported by others for COVID-positive patients. But we really wanted to dig into a little bit more of this inflammatory epithelial cells. Ni Huang in our lab carried out differential gene expression and asked and looked for genes that are upregulated in COVID positive versus negative cells. And you can see he found a panel of genes here. And when he used this group of upregulated genes in a GO-term enrichment analysis, the terms that came up were antimicrobial humoral response, then here neutrophil aggregation, neutrophil chemotaxis, neutrophil migration. So again, it really appears to be a cell type that is enriched and has a pivotal role in that neutrophil recruitment that we see. And then actually last week in Cell, there was a very nice paper that showed that calprotectin is actually a biomarker that can discriminate severe from mild COVID disease. And actually the protein calprotectin is a dimer of S100A8 and S100A9 that is one of these upregulated proteins. And of course, these are also very classical inflammatory markers that are often expressed in many cell types, macrophages, for example, which is why we call this an inflammatory epithelial cell type. We were also able to use this data set to look at viral reads. Again, as other people have reported, for many COVID-positive patients, we actually didn't see any reads at all. So this is the total viral reads in patients. There were 2 that had incredibly low levels, but then there were also 2 that really had quite high levels. And we then used the reads that we saw here and plotted the proportion of cells with viral reads in only the COVID-positive patients. And we see a confirmation of our ACE2 expression in that the secretory 2 cells, which are a club type cell, seem to have the highest level, but there's also low level in many of the other epithelial cell types. Here's some, here's some basal and some ciliated cells. But interestingly, we also see really quite high levels in inflammatory dendritic cells. And again, this makes sense in the biology because these are cells that take up virus in order to do antigen presentation. But actually, we also see a fair bit, not a large amount, but we do see viral reads across a number of different immune cells. Here in CD8 and NK cells, then here in mast cells, Langerhans cells, and so forth. So at this stage, we're really not sure how the virus gets in, but this is in vivo data, and we have faith in it because we know that the false positive rate is very low since COVID negative samples have never given any reads. And actually, I think with that, I think my time is up. And it just quickly go over the conclusions. We've generated a large single cell data set. We've identified a novel inflammatory epithelial cell state that is associated with COVID and that appears to be relevant in generating the antimicrobial response and neutrophil recruitment. And we've been able to detect viral reads in a range of different cell types, epithelial and inflammatory dendritic cells, and also at low levels in different immune cells. And all of this work was carried out at the Sanger Institute and at UCL. Here's the lovely team in the Teichmann lab that have contributed. And again, a great thank you for Masahiro and Kaylee, who've done all the work for the sample collection. The work I started to talk about at the beginning came from this publication and was very much a collaborative piece of work from the HCA Lung Biological Network. And then lastly, I'd like to thank all the funders listed and the patients and their families. Thank you.
Thank you, Kerstin, for an excellent talk. It's very interesting subject, and it's great to see that the HCA Lung Biological Network actually was able to contribute to this research in a meaningful way. There's some questions from the audience, and I'll wait until a few more come in. I first have a question myself. So if you compare SARS coronavirus 2 to SARS coronavirus 1 using the same sort of receptors for cell entry, how do you explain the difference? Do you have any explanation for that in the effectivity?
No, I think just on the expression rate data, we don't have that explanation, because obviously, as far as we know, they use the same receptor and we're using the same data. But I think there is data to say that the SARS-CoV-2 differs in the spike protein and therefore might be cleaved by different proteases. Furin, for example, which is another protease that has quite wide tissue distribution. So that might help. Of course, these are cell surface proteins, so they're also glycosylated. So there may be many other levels of regulation that we don't see at the transcriptional level.
Excellent, thanks. And then from the audience, there's a question: was patient race or ethnicity taken into account when sampling in this study?
Yes, so we have ethnicity data probably for about half the patients. The patients have to agree to have their ethnicity data recorded. And actually, all the patients come from a London cohort, which is actually ethnically very, very diverse. I think at the latest cohort, 40% of Londoners are non-white. And actually, we would really like to extend this cohort to really increase the proportion of Asian and African individuals, so that we get a much broader range of really what's normal, really for the human cell atlas, because obviously ethnicity makes a big difference for COVID. And interestingly, one of the patients in this data set that had very strong neutrophil infiltration was of African origin.
So one final question before we move on to the next speaker. Another question from the audience. So do you see or expect changes in the number of these inflammatory epithelial cells between children and adults?
And this is where I sort of had my disclaimer that this very early data analysis. At the moment, all of our adult data is COVID, whereas the children's data is both a mixture of normal and COVID. Now, the statistical model that we've used should account for that. But, you know, I think the answer is that we should really integrate our data with the normal nasal data set, for example, the one that Pascal Babry has generated, and then redo the analysis. And I'll feel much more comfortable to answer that question. But at the moment, it looks as if the inflammatory state is more frequent in children and less frequent in adult, but upregulated in both pediatric and adult COVID cases. But, you know, take it with a pinch of salt.
Okay, will do. Thanks, Kerstin, for this excellent talk.